Hey there, science fans! Today we're diving into the amazing world of DNA. It's the blueprint of life. It holds all the instructions to make you, you. Now imagine wanting to study just one tiny part of that massive blueprint. That's where PCR comes in. PCR is like a DNA photocopier. It lets us make billions of copies of a specific DNA sequence. This technology revolutionized science. It's used everywhere, from fighting crime to curing diseases. It's time to explore this incredible technique that's changing the world, one DNA copy at a time. Buckle up, science fans. So, what exactly is PCR? PCR stands for polymerase chain reaction. It's a bit of a mouthful, I know. Just think of it as a way to amplify DNA. We're talking making millions, even billions of copies of a specific DNA segment. Why would we want so many copies? Well, imagine trying to find a needle in a haystack. That's what it's like trying to study a single gene in your entire genome. PCR makes that needle much easier to find. It's a game changer for all sorts of fields, from medicine to forensics. We'll get into the specifics later. But first, let's meet the rock star who invented this amazing technique. The mastermind behind PCR is a guy named Kary Mullis. He was a bit of a rebel, a real free spirit. While driving his car one night in 1983, he had a flash of genius. He figured out how to copy DNA in a test tube. It was a revolutionary idea. Mullis won the Nobel Prize in Chemistry in 1993 for his invention. PCR is a real game changer, thanks to this brilliant, unconventional scientist. Now, let's get down to business. What do you need to perform this DNA copying magic trick? It's simpler than you might think. First, you need the DNA you want to copy. This is like the original document you put on the photocopier. Then, you need primers. These are short pieces of DNA that act like bookmarks. They tell the copying machinery where to start and stop copying. Next is DNA polymerase. This is the star of the show. It's an enzyme that reads the DNA template and adds matching DNA building blocks to make a new strand. Lastly, you need nucleotides. These are the individual building blocks of DNA. Think of them like the ink cartridges for our DNA photocopier. Mix them all together and you're ready to rock and roll. Section 5, Step 1, Denaturation. It's getting hot in here. PCR involves repeating a cycle of 3 steps. The first step is denaturation. We heat things up. Don't worry, we're not making DNA soup. The high temperature, around 94 to 98 degrees Celsius, causes the 2 strands of DNA to unzip and separate. It's like unzipping a zipper. Now we have 2 single strands of DNA ready for the next step. Section 6, Step 2, Annealing, primers on point. Time to cool things down. In the annealing step, we lower the temperature to around 50 to 65 degrees Celsius. This allows our primers to attach to their specific target sequences on the single-stranded DNA. Remember, the primers act like bookmarks, showing the DNA polymerase where to start copying. They're essential for making sure we're copying the right section of DNA. Section 7, Step 3, Extension, Building Blocks of Life. Now, for the main event, extension. We raise the temperature again to around 72 degrees Celsius, which is the perfect temperature for our DNA polymerase to work its magic. It starts at the primer and then adds matching nucleotides to the single-stranded DNA, making a brand new complementary strand. At the end of this step, we've doubled our DNA. We started with 1 copy, and now we have 2. Section 8, PCR's greatest hits from crime scenes to cures. Now let's talk about why PCR is such a big deal. This technique is used in countless applications. It's like having a superpower in the lab. Medical diagnostics PCR can detect tiny amounts of viral or bacterial DNA, helping diagnose infections like HIV and COVID-19 early on. Forensic science. Remember that needle in the haystack? PCR can find a single DNA molecule from a drop of blood or a strand of hair, helping solve crimes. Research scientists use PCR to study genes, create genetically modified organisms, and even trace ancestry. Agriculture PCR helps detect plant diseases, ensuring healthy crops and a more sustainable food supply. The possibilities are practically endless. Section 9. Encore. Advanced PCR techniques. Wait, there's more! Scientists are always innovating, and they've developed even more advanced PCR techniques like qPCR or real-time PCR. This technique allows us to measure the amount of DNA being copied in real time, making it super precise. It's like watching your DNA photocopier count the copies as it makes them, and then there's dPCR or digital PCR. This method divides the DNA sample into thousands of tiny droplets and performs PCR in each one. It's incredibly sensitive and can even detect rare mutations. PCR is a constantly evolving field, and these advanced techniques are pushing the boundaries of what's possible. So, there you have it. PCR is an incredible tool that's changing the world. It's helping us understand life, fight disease, solve crimes, and so much more. I encourage you to explore the fascinating world of PCR and see how it's shaping the future. Stay curious, science fans.